So hi everyone, this is Dr. Brown, and this is a session where I'm going to talk through how to analyze ELISA data. So this will follow on from the inflammatory biomarkers lecture that I have uploaded to Moodle, and this is going to be on how to handle, process, and present ELISA data. So the data that we're going to be working with is actually real data. So this is from a study that will look familiar, and it is something that we've covered in one of the very first Journal Club sessions. So this is from a paper or from a study that led to a paper from Johnston et al. So Will, who will help me in the live session next week, he basically wrote the paper, which you'll be familiar with, looking at these two treatment modalities for periodontal disease. And basically the data that we have is protein assessment of the gingival curricular fluid, the GCF. And basically the GCF was taken from patients with periodontal disease. And those patients received two types of treatment, so hand scaling versus ultrasonic instrumentation. The data that we'll work with and we'll work through in this session is for interleukin 8. So interleukin 8 is a pro-inflammatory cytokine, whilst you will be given data for an ELISA plate that is looking for TNF alpha. So TNF alpha is also a pro-inflammatory cytokine. The data that you'll be working with will contain 4 samples that are run in duplicate per patient, and there'll be a total of 10 patients used on the ELISA plate. And these 4 samples basically equate to multiple time points. So if you remember from the paper, basically the assessment of the protein levels was done at baseline, day 1, day 7, and day 90. So basically set 1, 7, and 90 days post-treatment. So as I've said, there's a total of 10 patients. The first 5 patients are from the hand scaling group, and the second 5 patients are from the ultrasonic group. So this should make a bit more sense when we actually work through the data in the coming slides. So this slide just shows the schematic for the study. Again, as I've said, this should be familiar to you. And this is a paper that we discussed in the very first week of Journal Club from our research group. And this is the study on the left-hand side, where you have 37 patients with periodontal disease. Essentially half of them received hand scaling and half of them received ultrasonic scaling instrumentation treatment. And this was done obviously at baseline, day 0, if you like. And then 1, 7, and 90 days after that, the patients returned, and the study was, or the design of the study was to basically look at the systemic inflammatory response. And this was specifically what was shown in this paper here. So basically the authors looked for 2 pro-inflammatory cytokines and also CRP, which we talked about in the inflammatory biomarkers lecture. The current work that they are working on, the current data analysis that the authors are working on, and specifically Will, he is looking at the microbial response. So he's basically trying to assess what the changes are to the microbiome following the 2 treatments. And then finally, he's also trying to look at not only the local clinical response, but also the local inflammatory response. And it's ultimately this local inflammatory response which is what the data set is on. And it's basically the ELISAs are looking at the cytokine response, as I've said, within the GCF of these patients, and specifically the 2 proteins that we're interested in is IL-8, which we'll walk through and work through in this session. And then the data that you'll get given is for TNF alpha. So as I've said, this is an actual real study, a real experiment that's been done. And basically this is what the ELISA plate looks like. So obviously we talked about the principles of ELISAs in the previous session. And this is essentially what a picture taken just on a phone of what the ELISA plate looks like after the color has developed. So on the left-hand side, the first 2 columns are the standards. So again, we talked about that in the pre-recorded lecture, where basically the very dark blue color is the highest concentration of interleukin 8. And what you see is this gradient of color going from a dark blue color down to a very light blue color. And these standards are in rows A to G, and then in row H, and specifically H1 and 2, these are your blanks, so there's no sample in these. And then all of the other wells contain your clinical samples. So there's a total of, as I've said, 10 patients, 4 samples for each patient from different time points, and all of those 4 samples are run in duplicate. I've got a plate plan in the coming slides, so hopefully that will make a bit more sense when I show you the plate plan. But this is essentially what an ELISA plate will look like once you've completed it. Of course, you need to actually get some sort of quantitative measure from an ELISA plate. And to do that, what we use is a microplate reader. And this is the name of the microplate reader that we use. So Fluorostar Omega microplate reader. And we specifically use the software that comes with the microtiter and the microplate reader to basically analyze, or at least to read our plate, and also to initially process the data before we can export it into Excel. So this is just a screenshot of the software once the plate has been read. So the plate has been read at 450 nanometers and also at 570 nanometers. And what you will see in the data here is the top number is basically the absorbance of each individual well. So this is a 96 well plate. Each individual well will have an absorbance value at 450, and this is the top number here. And then the bottom number is the absorbance at 570. So we read the absorbance at 570 just to basically background correct the change in the color that you associate with the ELISA. And that is essentially it for the first thing. So this is what we see when we read the plate. And the first thing we need to do is export into Excel. And of course, I've done this for you. So this is done on the machine in the dental school. And I've done this for you. But basically, this is all you see. And then you're able to export that, export the raw data into Excel. The first step when you get to Excel is, and what you will see in Excel is something that looks like this, okay? And basically, what you see here is that you have a 96 well plate layout that has the absorbance values at 450 on the top, and then a 96 well plate layout that shows you the absorbance values at 570. And as I've said, these absorbance values at 570 will be essentially the background readings, and the first thing we need to do is background correct our values. And so to do that, you basically have to subtract the values at 570 from the values at 450 for each individual well. So that is the first step that you will need to do, is basically make another 96 well plate layout here in Excel, and essentially minus the value from A1, if we use A1 as an example, the absorbance value at A1 for 570, and subtract that from the value at A1 for 450. And if you do that for every single well, then you will get a 96 well plate layout that looks like this, that has the background corrected absorbance values. Okay, so hopefully that makes sense and you are following at the moment. So the third step is to color code your data. And to do that, I would suggest to color code the background corrected values and also make an extra plate plan, an extra 96 well plate layout that includes the plate plan. And this is essentially what the plate plan will be for the plate that we're going to work through, and also for the data that you're going to get as well. So the plate plan is the same. And plate plans are really important in science because they basically can tell you what is in each well. And of course, if you're running multiple plates, then it is really important to keep on top of the plate plan so that you actually know what is in each well, because it can get quite confusing if you're running multiple ELISAs with slightly different plate plans. So that is the third step, is to color code the plate plan and color code each well, specifically what is in each well. And this is what is in each well in our samples. So we have standards in the first 2 columns and specifically up to row G, okay? And these are the standard concentrations, so 2000 picograms per mil all the way down to 31.25 picograms per mil. In wells H1 and H2, these are your blanks. So these are wells that contain no sample. So this will allow us to blank correct all of our absorbance values. And then all of the other wells contain our samples of interest. So these, as I've said, are 4 per patient, ran in duplicate, and there's essentially 10 patients in total. And these 4 samples per patient will be those at baseline, those at 1 day post-treatment, those at 7 days post-treatment, and those at 90 days post-treatment. And what we have is basically the first patient is the first 8 wells, the second patient is the next 8 wells, and so on across the plate. So this is the next step. So once you've color coded your plate plans, and essentially when you know what is in each well, then you're able to proceed and blank correct your values. So we've already background corrected the values using the absorbance values at 570. So that is important to do first. We also need to blank correct them. So blank correction is important because there will always be some residual color change occurring, even when there is no protein within your well. And as you can see here, there is an absorbance values of around 0.1 absorbance values at the OD of 450 following correction at 570. So basically there is some residual blue color within your wells, even when it contains no samples. So we need to take that into account for all of the wells. And to do that, what we do is blank correct them all. And essentially what you have to do first is average the blanks. So we always run them in duplicate. So average the blanks, and then you can do this at the bottom here. Basically take an average of the H1 and the H2 wells, or the absorbance values of H1 and H2. Once you've averaged the blanks, you can then use that number and subtract that number from all of your wells. Okay. And essentially, you just need to do that for every well, including your standards as well as your samples. And then the final step for the data analysis within Excel is to average the duplicate readings of your samples. You don't need to do that for the standards, and I'll explain why in a few minutes. But basically, you need to average the duplicate readings for each sample, and essentially that will allow you to just have one absorbance reading for each sample at each time point. And that is, of course, for each patient. And ultimately, what you will have is 40 values in total, 4 for each patient. Each of those 4 will be at a different time point. So moving on from using Excel into using Prism. So the next job that we need to do, so you should have a plate that a plate plan or a 96 well plate layout that looks something like this. So these will be background corrected and blank corrected values, and also averaged values for your samples. What you need to do first is open up GraphPad Prism. And what we want specifically is an XY table or an XY graph. And this should be the first thing that you see when you open GraphPad Prism. And what you need to enter is, or what you need to click on is enter in replicate data. Once you've clicked on that, you can then press create table or create graph. The next job, or should I say the second job when it comes to GraphPad Prism is that you need to essentially delete all of the unwanted columns. So I recall that Prism will give you sort of example data, and you need to ultimately sort of tidy up your GraphPad Prism file so that it only looks something like this. Okay, so you can delete group B from the example, change the number of replicates from 3 to 2, and then also correctly label all of the X and Y columns. So this should be second nature to you now that you've done a lot of data analysis over the last few months. So this should be second nature to you. Just make sure that everything is labeled correctly. And of course, for you, this probably isn't that important, but in the long term, if you want to be a good scientist, a good PhD student, or good research student, then you need to be able to correctly label all of your different files, because if you're working with multiple ELISA plates, then it can get quite confusing if you don't correctly label them. Once you've labeled them, you need to then copy and paste your data into the PRISM tables, and it should look something like this. So as I've said, we know the concentrations of the standards, so you need to include those. And then what you need to do is copy and paste the duplicate readings for your standards. So we haven't averaged them because we want to create a standard curve that has error bars. And basically what you need to do is copy and paste those values into GraphPad Prism. So it should look something like this. And then below those values, what you need to do is copy and paste all of your individual absorbance values for each patient and each time point. And all you need to do is copy and paste them below the standards. So you don't need to label anything in the X column, because of course we don't know the concentrations of those before we can calculate them using the standard curve. So you just keep the X column blank for these clinical sample, or the absorbance values of these clinical samples. So it should look something like this. Hopefully it should be second nature to you. You've had enough experience with GraphPad Prism. If you was to click on the graph, so of course you've made your data table. If you was to click on the graph, it should look something like this. Slightly different because of course you're being given a different data set. But as you can see, there's actual error bars within this graph. What I would recommend doing is changing the x-axis to log 10 scale so that you can see a nice sigmoidal shaped curve. Okay, you can keep the y-axis the same, but if you change the x-axis to log 10, then that will allow you to actually see this nice S-shaped curve of the standards. So the next step, so step 3, is to go and analyze the data. And specifically, you need to click on the Analyze button when you are on the data table. So this should be in the top row of icons. And then the first thing that you need to then do is press this option here. So this is interpolate a standard curve. And this is important because this will allow you to calculate unknown concentrations of your clinical samples using the standard curve. So once you press OK for that option, what you should see is a box that looks something like this. And as I've talked about in the inflammatory biomarkers lecture, what you will see in ELISAs is that standard curves for ELISAs are generally sigmoidal in shape, and we saw that in the previous slide. So they're kind of S-shaped. There's not a linear relationship between concentration and absorbance. So for that, and to take that into account, you need to click this option here. So this sigmoidal 4PL X is concentration. So that's the option that you need to pick. And that's basically just a mathematic model that applies a standard curve to your data set. So once you've pressed OK, then the next step is you should be able to then see your concentrations of your unknown samples in relation to their OD. And to do that, what should have popped up is an option here in the results section, which was basically the interpolation of the IL-8 ELISA. And what you will then see is basically all of your OD absorbance values that you've put in the y-axis in the data table. But what you will see here is that in the X column is that all of the absorbance values have basically been turned into concentrations of your given protein. And if you can scroll down in this, you will be able to copy and paste these, and essentially you are free now to present this data any way you wish. So we obviously know of the different clinical samples. You have different treatment groups, you have different treatment times, you have different patients. So it's very easy to go away and present this any way you like. Okay, so you can compare treatments, you can compare treatment times, you could actually present individual data for each individual patient. Very much depends on what you want to do. It just will give us an idea of whether you've understood how to handle ELISA data. And hopefully if you just follow the steps that I've provided here, then that should be quite self-explanatory. Okay, so that brings us to a close for the data analysis of the ELISAs. Hopefully, as I've said, that should be quite self-explanatory, and if you follow that process step by step, then you should be able to get some nice data at the end, and then you have a clean slate to basically go away and present that data any way you wish. So if I just get up the Excel sheet that contains the data that you will be working with, I'll just share my screen again. So this is essentially what I've taken the screenshots of. Unfortunately, you won't be receiving this. All you will be receiving is this here. So you will have absorbance values at 450. So remember, this is TNF alpha and not IL-8, even though you follow the exact same steps. You will have absorbance values at 450 and absorbance values at 570. And if you follow the same steps as I've shown in the PowerPoint, and as you can see here, where you background correct them, you make sure you have a nice labeled plate plan. You then take the averages of the blank and blank correct your samples, and then this data now can be copied and pasted into Prism. So if I just open up Prism so that you can see what I'm looking at and what I screenshotted for the PowerPoint. So if I just share my screen again, this is essentially what you will see. Okay, so you'll have in the Y columns, you'll have your two duplicates of your standards. Standards will be the exact same concentrations for TNF as they are for IL-8. And then basically all you've got to do is copy and paste all of your absorbance values from your ELISA plate into the first Y column. Once you have done that, you can check that your ELISA standard curve looks okay, and here it does. So as you can see, there's got a nice standard curve, and this is what it will look like once you've clicked on the S sigmoidal, so the 4PL S sigmoidal curve, and when you've analyzed the data. And then if you go to interpolation of the ELISA data, then essentially what you'll see is the concentration of your unknown samples for a given protein. So in this case, it's IL-8, and what you'll see is that each of these absorbance values essentially equates to a different concentration of the protein. So that brings us to a close for the session. As always, if anyone does have any questions or queries about the data analysis or about the inflammatory biomarkers lecture, then please do not hesitate to get in touch with me. And I will see you next Tuesday for the live session, where myself and also will be jumping in and out of breakout rooms and just checking that you understand how to handle and process ELISA data. As I've said, if you follow the process that I've just talked through, then it should be quite simple. Okay, thank you for your attention, and I will see you all next week.